Hello, I'm talking to you. This is Goddess of Science Biology, and I'm Mrs. Bolvin, and today we're going to talk about apoptosis and stem cells in part 6 of the Cellular Reproduction series. Okay, let's go. So, apoptosis is kind of an interesting thing. It's programmed cell death. This is a very interesting concept because this is one of the ways that last part we talked about cancer. This is one of the ways that if there's something wrong with a cell, if it's broken, if it's worn out, whatever, and at the end of its life, this is one of the ways that cells will self-destruct, so to speak. So what happens when an animal cell or a protist cell is there's something wrong with it, it's broken, or there's another instance, and I'll talk about that in just a minute. When the signal is given, it triggers the lysosomes, and we remember back from chapter on cell organelles that lysosomes contain a whole bunch of digestive enzymes. Triggers the lysosomes, they all burst open, digest the cell, and all those components of the cell are now ready to be used. The molecules and atoms are used by other cells, or they're disposed of as waste products. So the cell basically digests itself, which is a little bit weird. And I mentioned about worn out cells or cells that are broken or something like that. It's actually a natural part of development in a lot of different organisms. You probably at some point in your elementary science career just learned about the life cycle of a tadpole. It starts out as an egg, and then it's a tadpole fish-like thing, and then it loses its tail and turns into a frog. But there's that weird, awkward intermediate stage where it has both legs and a tail. Very weird. The way it gets rid of its tail is the cells that connect the tail to the body trigger their lysosomes, and so it digests itself and goes away. Same thing happens between your fingers and toes when you're inside your mom. Before, at a certain stage, all of your fingers are webbed. Your fingers and toes are webbed. And when the signal is given, those lysosomes and those cells burst, and it digests away a lot of that material. Now, some people continue to have webbing into, after they're born and things like that. There are abnormalities. Sometimes the lysosomes actually don't work, but that's how you get separation between your fingers and your toes. And a lot of times it's used for damaged cells, so damaged cells don't continue to reproduce and pass that damage on. Hopefully, the lysosomes work, but again, lysosomes are only in animal cells and protist cells. The other thing that we're going to talk about is stem cells, and there's a lot of great possibility in science surrounding stem cells. Stem cells are cells in an organism that don't have a specific job. Initially, we talked about a lot. You started out as a single cell, which was made when Sparky the sperm and Olivia ovum got together on a date, and they fused their nuclei and they became one cell called a zygote. That cell had all the information it needed to run every single cell in your body. So after a while of just regular these, like, it's any cell, it could become any cell. There's a lot of divisions that do that. Eventually, different cells are going to have to become muscle cells or liver cells or brain cells or something like that. So that is a very interesting process. Stem cells haven't gone through that process. Stem cells still have the potential to be any cell at all, which is really great. There are two different types of stem cells: embryonic stem cells, which are the early divisions of an embryo, of the zygote. It's after fertilization. It's a regular cell. It has all the information, but none of the genes have been turned on that, like, a liver cell would need or a skin cell would need. So it's about the first 100 to 150 cells that are made after the zygote continues to divide. And eventually they differentiate getting a job. When I worked in Boston at the Eye Research Institute, what we would do is we were working on retinal transplantation, and your retina is like a movie screen of neurons in the back of your eye, which is how it gathers the incoming light and converts that into an electrical signal or chemical signal that goes to your brain, and you say, ah, there's a spider over there, or oh, what a pretty flower. So it's part of your central nervous system. The problem with if you damage central nervous system, it doesn't really regenerate. It won't heal itself for the most part. So what we were doing is we were taking embryonic retina from rats and rabbits, and we were injecting that into adult rats and rabbits who had a very, very, very tiny incision made in their retina. It was very interesting. Anyway, so what we were trying to do is to heal that place that had been damaged with some embryonic retina, hoping that the surrounding cells would give the signals to the retina, the embryonic retina, that this is what you need to become. So they continued on with that, and many years after I left the lab, they had, I saw in the journals, the scientific journals, that they had actually done human trials. So that was quite amazing. So embryonic cells can be, different. You can differentiate them into different things. You could grow a liver. You could, all kinds of different things. Adults also have some stem cells in their body tissues, and these are usually for maintenance and repair. Also, like your blood cells have stem cells because you're constantly making more red blood cells, making white blood cells. There are stem cells that exist even in the adult organism just because we have to replace them so often. But the adult stem cells are really very, very limited as to what they could potentially become. Embryonic stem cells can become any tissue in the body. Adult, more limited. A couple of years ago, I saw a researcher had actually figured out how to take these adult body cells and turn them back into embryonic stem cells, like going back in time, like deprogramming them, which is really amazing, because they're looking at using a lot of stem cells to cure like Parkinson's disease and other really debilitating, spinal cord injuries, all kinds of stuff like that. They're looking at embryonic stem cells, but they're very limited in how many you have to do the research lines on those stem cell lines. So if they can take regular old adult body cells and turn them back into embryonic stem cells, that's huge. That's a giant thing. Okay, well, that's it for this time. Thanks for joining me. Have a great day.